Okay, you've been doing a lot of math. Let's check and make sure that you did all the math correctly, and then we'll move on to the interpretation. I worked out this group before in the last video. For the next group, what you should have gotten were expected values for 23, about 47, and 24. Those clearly are the opposite pattern that you saw in group A. We've got an unusually high number of heterozygotes in group B and a usually low number of homozygotes. For group C, the numbers are a little messier, but the pattern is the same as in group B. You've got more heterozygotes in this group than you would expect and fewer homozygotes. For group E, the numbers that you get are almost exactly the numbers that you expected to get. In other words, this population does seem to be at Hardy-Weinberg equilibrium. What do these data mean? Well, we'll be talking about that in our next class period. Right now, pause this video and write down what you think these patterns actually mean. Remember that there are 5 forces that can push a population away from Hardy-Weinberg equilibrium, although it should be fairly obvious which of them is involved when you have an epidemic. At the end of this video, there'll be an ungraded quiz that will ask you what you think is going on in this population. Go ahead and send in that answer, and we'll talk about it in our next class. So the case study we've been looking at, the Kuru epidemic, is a real-life situation in which researchers used Hardy-Weinberg equilibrium to figure out what was going on genetically in a population. But Hardy-Weinberg equilibrium can be used really pretty broadly to answer a lot of different questions. Whenever you see a departure from Hardy-Weinberg equilibrium, it doesn't just tell you, hey, something evolutionarily interesting is happening in this population, but it gives you as a clue about what that something interesting is. So for example, Hardy-Weinberg equilibrium is something I actually used in my research. I study mayflies. They're these unusual insects, and they're unusual because they're extraordinarily short-lived. When an adult, like this adult female, emerges as a imago or mature, she has at most a few days to live. Some mayflies, their adult lifespan lasts only a few hours. And during that time, she has got to reproduce, or that entire life was a waste. Sometimes it can be hard for a female to find a mate. For that reason, apparently, mayflies have evolved to have a backup emergency option. This female can mate with a male and lay eggs if everything goes well, but if she can't find a male to mate with and her time is running out, she can essentially self-fertilize. What that means is that she essentially mates with herself to generate diploid eggs that can then grow up to be viable offspring. How is it possible to tell if these mayflies are doing this? Well, catching them in the act of mating or not mating in this case is incredibly difficult, logistically speaking. It's just not practical to try to do it. But what you can do is collect a bunch of immature mayflies from a stream and look at their genotypes. Self-fertilization is basically the most dramatic form of inbreeding, or positive assortative mating. And when you have positive assortative mating, you get a higher frequency of homozygotes than you would expect in a population. So by looking at allele frequencies and genotype frequencies in a population, I was able to determine that yes, in fact, it did look like mayflies are reproducing asexually in some of the populations I study. That leads to other hypotheses about why they're reproducing asexually in some places and not others. But a comparison to Hardy-Weinberg equilibrium was a critical step in moving this research forward towards figuring out what's going on here. Looking for departures from Hardy-Weinberg equilibrium isn't the best way to detect all 5 of these evolutionary mechanisms here. Mutation is such a weak force by itself that it essentially never changes allele and genotype frequencies, not to a degree that you can actually detect. In the real world, data is always noisy, and mutation is such a weak force that its effect simply gets swamped by noise. Departures from Hardy-Weinberg equilibrium can tell you that maybe you should be looking for genetic drift or gene flow, but they're actually much more sensitive tests for these mechanisms. However, departures from Hardy-Weinberg equilibrium are, in and of themselves, a good way to check and see if non-random mating is occurring. Conservation biology is a branch of biology that is behind conserving, or that is protecting, endangered or threatened species in the wild. And many times, because these organisms are endangered or threatened, you're talking about populations that are very small. Whenever you get small populations, one of the things that conservation biologists are concerned with is that it may begin inbreeding. And as I was just discussing with the mayflies, inbreeding shows up as an unusually small frequency of heterozygotes. You get too many homozygotes and you get too few heterozygotes compared to what you'd expect from Hardy-Weinberg equilibrium. So for example, researchers looking at this amphibian, the Sonora tiger salamander, looked at the genotypes of a number of different populations and found that they were reliably missing a lot of heterozygotes. This suggests that a lot of inbreeding is going on. And because inbreeding leads to individuals that may be in generally poor health, that suggests that this species needs more protection, more rigorous protection, and needs more care invested in it to keep it from going extinct. If they found that inbreeding was not happening, it might suggest that you're dealing with healthier individuals that did not need as much effort to keep them from the brink of extinction. However, it's selection that Hardy-Weinberg equilibrium can really be used to look for. There are a lot of studies that have done essentially what you did in the case study about the Kuru epidemic. If an allele or a genotype is associated in some way with a disease, either positively or negatively, it won't be at Hardy-Weinberg equilibrium when you look at a group of individuals that have that disease, or if you specifically look at individuals who do not have that disease. This has been used in literally hundreds of published research studies that you see these days. For example, tuberculosis is the second deadliest infectious agent in humans today. You can see the effects of it here, these kind of grainy pictures of the lungs on an X-ray. That's because a tuberculosis infection has spread all through this individual's lungs. In 2013, researchers looking at tuberculosis found that individuals with the disease seem to be lacking a particular allele. And that leads to the hypothesis that this allele has some protective function against this very important deadly disease in humans. Conversely, another group that was looking at this gene here, the gene that makes the COMT protein in the nervous system, were trying to establish a link between this protein and schizophrenia. A few previous studies had suggested that it was possible that this protein was involved in schizophrenia. There's a known mutant version of this protein, and researchers had suggested that perhaps it was responsible for that particular disease. A study that looked at many individuals genetically found that this allele was at Hardy-Weinberg equilibrium in a group of individuals that had schizophrenia. And that strongly suggests that this mutant allele is not related to that disease. You can look at departures from Hardy-Weinberg equilibrium to test whether or not a particular genotype is associated with any kind of outcome, not just a specific disease. So for example, this and this are the same species of insect feeding on two different plants. The insect comes in two different color morphs. It should be pretty obvious that this one with a white stripe down its back is very well camouflaged against these leaves that have white stripes. But this speckled green one is much better camouflaged over on this host plant, which it usually inhabits. What some researchers did is they took large numbers of these insects and they put them on the wrong plant. So they put the striped insect over here on the speckled plant and the speckled insect over on the striped plant. Then they just left them there for 8 days and then came back and collected all of the survivors that they could find. Then they sequenced the genomes of each one of the surviving insects and looked to see if there were alleles or genotypes that were particularly associated with surviving for 8 days on the wrong plant. And they found a great many loci in the genome that did seem to be unusually frequent among the survivors. They found genotypes that were unusually likely to be found among survivors. It's true that a lot of these alleles they subsequently found had to do with color pattern, but there were a tremendous number of them that didn't. So it suggests that a lot of genetic traits in this insect, other than color pattern, help them to survive when they're in the wrong context in the wild. And that, of course, opens up the avenue for a tremendous number of other studies. Why am I telling you all this? Well, Hardy-Weinberg is one of the least popular topics in many introductory biology classes, largely because students don't see why it's relevant or interesting. It's not immediately clear what you do with it. However, especially today, when genomic techniques are making tremendous amounts of genetic data available, it's really easy for us to look for departures from Hardy-Weinberg equilibrium in the context of all sorts of different conditions and diseases. Hardy-Weinberg equilibrium is in fact a really powerful tool that you can use to understand what's going on in all sorts of evolutionary situations, provided you understand how to use it.